Here is the central dogma of biology. Now, perhaps you haven't heard this model called the central dogma before, but it is in fact the central tenet around which most biological pedagogy is developed. We start off with DNA, and not just DNA, but a gene on a DNA, a double helix of DNA. So a gene is transcribed, or perhaps a series of genes, by the way, is transcribed into RNA. And usually when we talk about RNA, we're talking about an mRNA transcript. The mRNA transcript is then translated into an amino acid sequence, which then in turn is folded into a protein. It always folds exactly the same way given a particular temperature and a particular pH. It always folds the same way. This is important. So again, the action words here are transcription and translation. DNA is transcribed into RNA. RNA is translated into amino acids. Amino acids are folded into a protein. And proteins are going to be coming up again and again in our class, where we're going to be discussing the role of proteins in certain physiological functions. It's going to come up again and again and again. So I want you to keep in mind that if something goes wrong at the level of the gene, for example, a mutation, it could cause, especially a deleterious mutation, could cause an erroneous protein folding. At the end of the day, this could cause a loss of physiological function.